Dr. Will Yates is an ophthalmologist and aspiring clinical scientist involved in basic science and clinical research. He's currently a clinical associate lecturer at the SaveSight Institute and completing his PhD under Professor Robyn Jamieson at the Children's Medical Research Institute, investigating the natural history, disease modelling and gene therapy for inherited retinal diseases. Will plans to travel to the US and Canada in 2025 for future pursue research, as well as clinical trial experience in inherited retinal diseases. Thank you very much, Will.
Thanks for the introduction, Mike, and thank you for the SaveSight Institute for inviting me along in John and Robyn's absence. It's nice to dovetail after Lily and Jenna. What we're working on is on inherited retinal diseases, and I'm hoping to represent the few ophthalmologists that hopefully have a little bit more tact in delivering news to patients and not treating people like they're textbook cases of RP. So I'm hoping I can instill a bit of faith back into our people. So as I mentioned, I'd like to acknowledge Robyn Jamieson and John Grigg, as well as the surgeons who deliver some of the gene therapy, Matthew Simeonovich and Gaurav Bhardwaj, and one of the postdocs in the laboratory that I work at, which is at the Children's Medical Research Institute, Tuhar Loy, who's taken me under her wing and taught an ophthalmologist a lot about science. So this is quite a busy slide, but inherited retinal diseases are now the leading cause of blindness in children and working age adults. And the prevalence is about 1 in 1,000, 1 in 1,500 people. And we know there's about 250 genes that result in inherited retinal diseases. And that's it. We're discovering more genes every time. Now, the majority come from autosomal recessive. So that's where you have a spelling mistake in each of the genes from each parent. And that's the majority. There's also the autosomal dominant form, which is where you inherit a spelling mistake in just one of the genes, but that spelling mistake actually interferes with the good copy, and that's autosomal dominant. And then you have the X-linked, which tends to affect the males in the group. And inherited retinal diseases can just cause the inherited retinal disease, so the retinitis pigmentosa, the cone rod dystrophy. It can cause the rod cone dystrophy or the macular dystrophy, but it can also be associated with hearing loss in Usher's syndrome and other different affects multiple different systems. And you can see that this is quite a busy slide, but the genes, it's really complex and all the genes overlap, and they can give different phenotypes, and the most severe being the Leber's congenital amaurosis, which affects children in their first year of life. But people like Lily can have pretty normal vision up until their mid-20s, and then they then slowly lose their vision. So it's a different journey for everyone. And now that we have a better understanding of the genetics, it's now becoming clearer, and I think we're now working towards therapies. So this is, again, a bit of a complex slide, but I just want to bring your attention to here, where what we're really focusing on is the knowledge of the genetic cause. And once we have the knowledge of the genetic cause, if we have viable or retinal cells that are still functioning, we can deliver a gene therapy that will hopefully correct the spelling mistake in someone's DNA at that level. But down the far end, we're now looking, there's multiple groups, including Matthew Simeonovich's group and Mark Gillies' group, working on therapies for people who've had really no functioning retina, about how you can then stimulate those cells, the remaining cells or the remaining parts of the retina, into being able to sense light again. So this is a really exciting prospect, and there's options right across the spectrum, and there's so many phase 1, 2 clinical trials, and we're progressing into phase 3, and we now have, as Peter mentioned, we've now treated 11 patients with LUXTURNA at the Children's Hospital Westmead, both children and young adults. So this is just a little bit of a guide to what we actually do with gene replacement therapy. So it's where we introduce a good copy of the DNA, but it doesn't change the person's DNA itself. And it needs to be delivered in a vehicle. And when I mean a vehicle, we're talking microns, we're talking nanometers, we're talking about millionths of a millimeter. And so the way to deliver that genetic material is actually in the form of a virus. And these are deactivated viruses that are totally harmless. And they actually hitch a ride. So we surgically deliver the gene therapy and they hitch a ride into the cell. And then that functioning copy of the DNA then tells the cell to start producing the protein that isn't working. And this all began with I'll give you the story of RPE65, which is what the gene therapy LUXTURNA is treating. So this gene was actually discovered in 1993, and the first FDA approval was in 2017. So we're talking a long time from discovery to now a therapy. And it actually started with a dog model in a Briard dog, and this is Lancelot the dog. And Lancelot the dog actually plays a very important role in trying to get government funding, Congress, the NIH, the Foundation Fighting Retinal Blindness to pay for this. I mean, so the dog model was quite interesting. These dogs, they were born, they had a little bit of vision, but by the time they were sort of 1 year old, they just couldn't see, they couldn't navigate. And so they followed them for about 10 years. And the gene augmentation therapies, so the gene replacement therapies that actually LUXTURNA is based on, was started in 2000, and the clinical trials extended up until 2008. And they were very expensive preclinical trials to run. I mean, looking after dogs costs $30 a day. You're doing that over 10 years. So they really needed to be able to demonstrate to the people paying for this, so the congressmen and the congresswomen, how to get funding into this. And so they took Lancelot down to Congress. That's Lancelot in front of the Capitol building. And they took Lancelot along with one of his litter mates. And the litter mate couldn't see, that hadn't had gene therapy, but Lancelot could. He was running around normally. And this was enough to convince the politicians to move forward on this and start funding a lot of the sort of phase 2 clinical trials. And then it got picked up by one of the startup pharmaceutical companies called Spark Therapeutics. And now it's been rolled out throughout the world by one of the major drug companies to enable us to now treat patients in Australia. So it was TGA approved in 2020, and at the Children's Hospital Westmead, we treated our first few patients there, and there's now been 11 patients that have been treated, and the age range is from 5 years all the way up to 45 years, and all the patients have been noticing an improvement in their vision at 3 months, and again at 1 year. And these are patients who now, so this is the preclinical, this is just some of the, this is, I'm hitting that, Emma. So this is, I'll just mute the American accent. So on the left here, this is the patient who is pre-treatment, so hasn't had gene therapy, and this is called a multi-luminance maze test. And this is what convinced the regulators. We're going to have to start from the beginning because they're kind of halfway through the maze now. And then this is 3 months post the test, and you can see this patient here is struggling to really navigate. And this is in a low luminance, and this is what retinitis pigmentosa patients experience. They can't navigate at night, they can't see the stars. And you can see they're really struggling, they're tripping over everything here. But you can see 3 months post-treatment, these patients are easily navigating the maze. So this is enough to get the Food and Drug Administration to approve LUXTURNA, and then subsequently the Therapeutic Goods Administration, or the TGA, to approve LUXTURNA therapy. And you can see it's just pretty remarkable, the improvement. The person on the left, pre-treatment, needed to be reorientated. It took them, they just never completed the maze, but the other person's already well and truly finished. Thank you. So the Sydney experience, you've probably seen this slide on Peter's talk. It takes a massive team. So this is really spearheaded by Professor Jamieson and Professor Grigg, who many of you have met in the audience. And then the two surgeons here are Matthew Simonovich and Guru Bhardwaj, but we've got orthoptists, we've got the pharmacists, we've got the expert nursing team, we've got a huge amount of people who try and helping deliver this therapy. And this is the first one, but I am envisaging over the next couple of years, we're going to have more and more therapies come online. The therapy actually gets shipped over from Switzerland, and it's stored at -80 degrees, so it takes a long time to get to room temperature to then be able to deliver into the eye. So now the next one is a surgical video of us delivering gene therapy. So look, the nice thing about ophthalmology is there's not a lot of blood. So, but just if you're a bit squeamish, I'm just going to give you a bit of a warning. So this is where we do keyhole surgery, and what we're doing is that's the retina there, and we've just injected some water between the layers of the retina. So this is 200 microns, and then we're using an intraoperative OCT. And what we do is that's a special device to look at the layers, and then we can deliver the gene therapy directly in conjunction with the cells where it needs to be. And it's an amazing technique, and I think this is going to revolutionize how we deliver gene therapies in the future. And more drugs and inherited retinal diseases are now becoming online. On my next slide, thanks. Oh, sorry, this was just a couple of the patients who, I think you may have seen this article in the ABC. They can now navigate, they can find their chair on the train. These were two young adults who couldn't find their chair on the train, but now they can look at the stars, they can navigate, they've got a little bit more independence back. We're not talking about probably getting driver's license back or anything like that, but we're talking about really functional gains for patients. Now, it's a busy slide, but there is so much going on in this area. There's currently, and this slide's probably a few months old, there's currently 125 interventional trials. So phase 1 is where we really try and move it from the preclinical stage into human. And then phase 2 is really safety. And then phase 3 is really how effective it is before you then push it into to the market. And you can see that there's so many people are working on choroideremia, X-linked retinoschisis, achromatopsia. There's heaps of work going on here. X-linked RP, the CEP290, which is another quite common inherited retinal disease. USH1B, which is one of those inherited retinal diseases that cause both blindness and deafness. There's some promising work, and that's recruiting patients next year in Italy and the UK. And then in phase 3, which is where we're kind of the closest to market, to getting it out to patients, is we've got RPGR, which is one of the more common inherited retinal diseases. And the hope is that we may be able to have some patients in Australia treated in the next couple of years. And then not strictly inherited retinal diseases, but Leber's hereditary optic neuropathy is a disease that affects the optic nerve. There's a really promising rescue therapy for these patients where we're being able to salvage some of their vision and actually get some improvement in their vision. So this is all really exciting in the realm of inherited retinal diseases. So I just want to touch on what we're sort of working on at the Children's Hospital Westmead, at the Children's Medical Research Institute and the SafeSight Institute. So we're looking at both sort of mouse models where we make a gene therapy in mice that have the same mutations. Humans, believe it or not, humans and mice share about 80% of the same genes, and they're pretty much exactly the same. It's pretty insane, but they're a good model to look at. And what we can do is, look, it's very hard to get a mice to tell you, is that clear or that's clear, or is this clearer? So we can't do a visual acuity like we do in humans, but what we do is we have these spinning grates, and we can be able to test their right and left eye to see if they can see. We can do OCTs, which is a very common, you've all probably been to the optometrist, so we can check their layers of the retina, and then we can do electroretinograms, which help us tell the function. And that's how we're testing our gene therapies and what we're working on in the mouse models. And then a really exciting prospect is these retinal organoids. Now, what we can do from a patient's blood sample with the spelling mistake in their DNA, we can actually turn those red blood cells, or those cells in their blood, and we can turn them into retinas. And we do that by sort of telling them to grow into induced pluripotent stem cells, which are like stem cells. You've probably all heard of stem cells. And what we do is then we can grow these retinas, and we can test our therapy on human tissue to see if we can rescue it. And we can see rods and cones in human tissue in culture and be able to look. And I think this is going to be a really promising way that we can test our therapies in a more humanized model as some of the research does shift a little bit away from animals, and we try and have a little bit more humanized research models. So Professor Jamieson asked me to just mention that, look, for those of you who have inherited retinal disease and don't have a genetic diagnosis, it's very easy to get linked into your genetic service. You can just go onto the genetics education website here, and you can type in your postcode, and that will tell you your local genetics service where you'll be able to have your genetics testing done free of charge. You'll meet a genetic counselor or a geneticist, and they'll be able to take the samples from yourself, the parents, and then be able to at least have as Lily said, we are finding answers for about 60% of patients, 60 to 70% of patients. And for those that we don't find answers to, we have a lot of tools at our disposal to try and find answers for those patients. And that's more in the research realm where Professor Jamieson works on. And so I'd just like to acknowledge the massive team here. I'm an ophthalmologist learning science. It's been a steep learning curve, but I've had excellent mentorship under Professor Jamieson, as well as Professor Grigg, as well as the postdocs in our laboratory. I also want to acknowledge Peter McCluskey for his mentorship for me since I was a medical student, as well as the surgeons in the group. And thank you all for your attention.
Go back. I have a suspicion there'll be some questions. Exactly. Okay. Lily's closest to me. Here's the mic.
Firstly, I just want to say thank you to SafeSight Institute. And, you know, when this is the first time since I've been diagnosed at 24 that I'm starting to understand my condition. And it's because you guys are sort of talking to us, you know, about what's going on in the back of the eye. I never knew that. So I just want to say thank you for days like today. Because I'm learning so much, and again, giving me so much hope. But I heard something about your growing eyeballs. Tell us about that.
Balls. I mean, we're growing retinas.
Okay.
And the retinas have the cones and the rods, which are the predominant cells that are damaged with retinitis pigmentosa, rod cone dystrophy, cone rod dystrophy. And we're also growing retinal pigment epithelium, which is the other cell type that gets diseased in inherited retinal diseases. And we're able to look at those and look at the spelling mistakes and be able to model those as a way to try and recapitulate. We'll look at what's going on at the cellular level, and we're actually testing our gene therapies on those as possible evidence to then try and move that into phase 1 clinical trials, which is probably a slightly better model than using mice and dogs and monkeys, which is where a lot of the research is done as well. So there is a little bit of a shift towards trying to do it, and it may be a little bit more of an accelerated way of getting therapies. Obviously, safety is paramount, but we think that this is possibly a nice model moving forward. And Dr. Anae Gonzalez Cordero, she's come from the University College of London, and she set up her laboratory at the Children's Medical Research Institute, and she's a world expert in growing retinal organoids from stem cells. Yeah.
Fantastic. Thank you.
This is just to know my son has cone rod dystrophy, and genetic test had identified RP2 genes as the cause. RP2. Yeah. So just want to know if any research is happening, and if so, in which of those stages it is.
I think RP2 is at preclinical still, so I don't think they've treated any human. They're not at the human clinical trial stage yet.
Okay, so it's in preclinical. Thank you.
In the O'Kane family.
G'day. So you mentioned there's about 200 classes of this type of disease. Now, when you develop therapies for it, can you do one and then change the parameters to get the other 199, or do you have to have 200 different treatments?
Currently, that's what we're working on. So I think the difficulty with this is I think what will happen is, I think each little, so the Australian, Chinese group, the American group, the Italian groups, the hope is that everyone works on their different genes and that we can work together as an international consortium. Obviously, that takes wriggle room with governments. This is expensive. I think the technology will continue to get cheaper. But I think at the moment, the holy grail is a gene agnostic approach where you'll deliver a therapy which will hopefully correct a lot of the genes, but we're just not quite there yet. So this is what we're working with. But at the other end, there are people working on therapies that will treat people that are really on the severe end with other modalities it doesn't matter what your spelling mistake is, and to see if there's a response. But at the moment, that's what we're working on. There's so many groups working on ABCA4, which is the most common inherited retinal disease gene. And the hope is that I think it's going to take a little bit of a mindset change from the FDA, the TGA, the EMA to go, well, look, we need to try and work together for these sort of rare diseases. Unfortunately, some of the issue we are finding is that it's expensive to get into a phase 3 clinical trial. You often need to get it to a drug company because they wear the risk and the cost of running a phase 3 clinical trial. And they just won't invest in it if they don't think it's a good business model, because they obviously have stakeholders, they have shareholders. So we need to be looking at alternative models for how to do this and deliver the therapies to patients. And the New South Wales government's invested about $180 million into building a gene therapy manufacturing facility on the Westmead Hospital campus, just behind the Children's Medical Research Institute. So the hope is that we may be able to do a lot of this stuff in-house within New South Wales and be a powerhouse of the biotech space.
Okay, I'm gonna. Over here. Here's the microphone.
Thank you very much. Hi, Will. Amazing speech.
Yes.
So yes, and I wanted to let you and everyone know that I actually got mine done just over a week ago at Westmead. So it's thank you. So thanks so much to the team, the nurses, the doctors, and everyone at Westmead were absolutely incredible and supportive, even to the point where they were looking after my guide dog and making sure he was there for my recovery. And yeah, it was just incredible. But yeah, early days for me still, but I can detect texture for the first time on certain fabrics, which is exciting because I love clothes. 2 questions for you, hopefully quick ones. That time period between 1993 and 2017, is that kind of, were they taking that whole kind of time period to develop the therapy, or was it like, okay, so we found a gene, now what do we do about it? And then there was a pause. And then my second question was, why RPE65? Why was that gene chosen first?
Yeah, good question. So RP65 is probably one of the more common inherited retinal disease genes. It was probably one of the earlier discovered ones, and they just had this dog model of which Lancelot came out of, which trying to deliver gene therapy in a dog is a slightly bigger eye than what I try and do, which is deliver it into a mouse, baby mice, our eye. And I think that the thing is, what traditionally we weren't really interested in because there was sort of no cures or no horizon of no prospect of cures. We didn't have a lot of the natural history. So we needed to know, well, what the natural history RP65 was, or what the different genes are. And so the natural history helps guide us, well, if we're going to deliver a therapy, can we change that natural history for the better for the patients? So we needed that, and that they need to do that in the dogs. That also happened alongside with the human stuff. And then also we needed to have a clinical endpoint that they could show to the FDA, which was the maze test, to say, this works, let us move forward with it. Yeah.
Yeah, fascinating. Thank you.
Thank you.
Awesome. So how's that for therapy getting real in the room? Anybody else? Who got in first? You're closer.
Hello, we're lucky that Rosie has donated her blood, so she's part of the Stargardt's ABCA4 trial. I had 2 questions. One was how long it takes to progress. Do you expect to progress from the blood collection growth stage through to a potential trial?
at the cell level or into a human trial?
To a human trial.
The cells, they're not perfect. It takes about 30 to 40 weeks to grow from Rosie's blood into photoreceptors that we can then trial test therapies. And then obviously we've got to do the cutting with the molecular cutting with what we do to make the therapy, test the therapies. Look, we're looking at sort of 3 to 5 years possibly. And then in the things in science, which I've learnt, is it's not like clinical medicine where things should work 100% of the time almost. Science is a journey, and if things work 5 to 10% of the time, you're doing well. So it just really depends. But there's so many people working on ABCA4 because that's the next big front. That's one of the next big frontiers. Yeah.
Thank you. And my other query was, are the genetic, the gen cell therapy trials, is the hope that they will reverse the condition or prevent further deterioration?
So some people are working on, so there's gene replacement therapy, which is where you're just trying to replace the spelling mistake and get the protein or getting those cells functioning properly. And that really works the best in people who still have functioning rods and cones or functioning retinas. There's people also working on stem cell therapy, and that's coming away. It's probably a little bit further behind, but stem cell therapy is where they're actually trying to replace those cells. And there's some really promising stuff in that field as well.
Okay, thank you very much.
Over here for maybe our last question.
My name is Irina Bruckner. I'm a vision teacher. I just wanted to ask, the test that you have shown there, is that the same standard test that's used in all countries? Or is it something that you have developed?
Testing?
No, the the maze test that was on a video.
Yeah. So pretty much most people who are doing any animal work, dog work, human work, do that sort of stuff where we want to look at visual behavior. Are we getting a response with visual behavior? Are we rescuing the retina without having to look at the retina under a microscope? And is the retina responding as appropriately? So that's quite well trodden. The retinal organoids that you grow from patients, this is a rapidly evolving field, and I think there will be a consensus and people having a sort of idea of, well, you need to be able to look at these things to know that you're having a rescue before we then think about taking this to a phase to human trials, first in human trials.
So each virus that you deliver to the patient's retina is custom made for each patient?
No. So the viruses, are pretty amazing. And again, that's some people's PhD and life passion is actually designing the viruses that will target the particular cell that you want to deliver the therapy in and not the others. So people are working on that. And by the time it's getting to phase just human trials, it's all pretty standardized. Yeah.
Thank you.
Thank you, Will. You're an exceptional communicator